Well, I appreciate the opportunity to share some of my thoughts and experiences with working in the field of nutritional immunology, particularly as it relates to inflammation. And there's a number of reasons why we can certainly imagine that inflammation and inflammatory biomarkers might be useful in setting dietary guidelines for dietary fats and some of the components of dietary fats. And we won't be necessarily diving into the specific evidence or literature for each of these points, but I wanted to try to provide a comprehensive overview of why inflammation biomarkers might be useful. And some of you may have heard or are aware of the fact that there's evidence out there that high-fat meals will induce a transient inflammatory excursion that occurs following the meal, and that there's some exciting novel evidence out there. Again, at this point it's only animal-based, that suggests that certain types of dietary fats can influence the gut microflora, and that this can subsequently impact our health, and particularly as it relates to inflammatory status. From a nutrient-based perspective, there's also quite a bit of evidence and literature out there that suggests that certain fatty acids, which are the major building blocks of dietary fats, so certain fatty acids may either promote or reduce low-grade inflammation, and we will talk a little bit about some of that evidence. And then we shouldn't ignore the fact that even though dietary fats are 97 to 99% triglyceride based, they do have non-triglyceride components, for us lipid chemists, the non-saponifiable fraction. And particularly when you're talking about Mediterranean diets and extra virgin olive oil, I think it's very important to recognize that high oleic acid vegetable oils may not necessarily recapitulate the health effects of high oleic acid extra virgin olive oil because of some of these non-saponifiable or non-triglyceride components that are in these oils. So that's important to keep that in mind. But the general public has been, to some extent, surprised by inflammation, okay? And about 10 years ago, Time had this cover, The Secret Killer. Well, for those of us in life sciences or biomedical research, the role that inflammation plays in our health has certainly been no surprise. It's not been a secret. And we clearly are trying to understand the underlying role that inflammation plays in a whole variety of diseases. And so you can just pick your disease of interest and find literature that suggests that inflammation is playing some kind of a role. But I also think it's really important to recognize that most of the attention has been on too much inflammation causing disease, and there's no doubt that that is true. But inflammation is actually a natural and healthy, normal, essential process that has evolved with us. And so you can equally say that zero inflammation would not necessarily be good health either, because without an inflammatory response, your immune system does not do its job, and you're, again, going to likely have higher mortality associated with that. And so I'd like to believe that at some place in there is this optimal level of inflammatory response that is necessary for good health. But the inflammatory response is very complex. It involves a whole host of different cells and tissues throughout the body. It also involves a large assortment of mediators. And it is these various soluble mediators that are typically the inflammatory biomarkers that we measure in blood samples or other tissue samples. And here's just a small list of some of the more common ones. And I wanted to point out that, again, it's not only some proteins, but sometimes there are receptors, and sometimes there are actual enzymes that are found in the blood that can be important as a biomarker for inflammatory responses. The other thing I'd like to point out, is this a laser pointer? Okay. Is, again, the attention for most of the research to date has been on this acute phase. Some some event, bacteria, trauma, shock, surgery, whatever, induces an acute inflammatory response, and then eventually that response hopefully comes back down to zero. And that's what we're now beginning to understand is that nutrition may be playing a much more important role at the resolution phase of this response rather than the acute elevation phase. And in fact, there's been some very exciting results coming out of a number of labs, including Charlie Serhan's lab here at Harvard, that has suggested that some of the polyunsaturated fatty acid lipid mediators may be playing critical roles in health by promoting resolution. And again, so resolution of inflammation is not just a passive process that just occurs once the trauma is over. It is actually an active process that we are just beginning to unravel. And again, I think this is an exciting area for the future, and it's where more nutrition research needs to be focusing their attention, not just on the front end of the inflammatory response. So if you look in the literature, you'll see lots of examples of the relationship between foods and inflammation. And it certainly seems as if we know everything that we need to know about this thing. And so again, all you have to do is go on Amazon and you can order one of these wonderful books and read about all of the things that we think we know or hope that we know. And again, in many respects, the perspective of these books is basically saying that, well, look, if you just eat the right foods, it will solve all of your health problems as it relates to inflammation. And I'm confident that we don't yet know everything that we need to know. I'm also very pleased that the ILSI group, the ILSI organization, and the groups in Europe have been really leading the way in this inflammatory biomarker area. And this report from 2011 is really an excellent, comprehensive report that touches on a variety of these topics that I think that if you are interested in this subject matter, I would very much recommend that you start your reading with this report, and you will find a host of useful information and excellent research, but also posing some interesting questions. I wanted to provide you early in this talk with an example of some of the kinds of data that exist out there in terms of the links between dietary lipids and inflammation. And this comes out of essentially the Nurses' Health Study, where a cohort or a group, a subset of the people involved in this study, they took blood samples and they analyzed them for inflammatory biomarkers, and they broke them up into quintiles in terms of their trans fatty acid intake. And what I want to point out is, again, from the lowest quartile of trans fatty acid intake, which is a little bit less than 1 energy percent of trans fats in the diet, versus the highest quintile, which is a little bit over 2 energy percent trans fats, you could see a dose-dependent increase in an inflammatory biomarker called C-reactive protein. And I want you to pay attention to the concentration, milligrams per liter, and they start out at about 1, and it goes up to almost 2 milligrams per liter. And we'll talk a little bit about what those values actually mean. But you can see that the N is about 146 nurses in each of these groups, and that the statistical trend for a significant correlation existed, and they adjusted, as all good correlational studies should do, is they adjust for potential confounding factors. And as it relates to inflammation, there is no question that you must control for age, body mass index, smoking, physical exercise, alcohol intake, and the intakes of different forms of fatty acids as well. In this case, since it was one of the key things looking at trans fats. And again, I believe that there's evidence to suggest that saturated fats, monounsaturated and polyunsaturated fats can influence low-grade inflammation, and so we have to control for that when we're looking for relationships. And so again, this 70% increase is statistically significant. I think the real important question here is it biologically meaningful? And that's a question that I can't answer, and I don't think that the rest of the experts in the field are really ready yet to answer whether or not these kinds of changes are important for our health. Another thing I wanted to point out is that oftentimes in these studies, researchers do not talk about how many of the subjects were actually below the limit of detection. And many of these biomarkers you'll find in healthy individuals that their actual concentration is below the limit of detection. And so that needs to be described in the methods very clearly, that if a certain amount of the individuals in your groups actually have no detectable inflammatory biomarker levels, then you have to describe, well, what did you assign them? Did you assign them 0, or did you assign them half of the lowest limit of detection, which is what's recommended? But you have to explain it. You can't just assume. So C-reactive protein is probably the most common inflammation biomarker, but if there's any single message that you get from this talk today, please, please get this message. It is not, in and of itself, an inclusive marker of inflammation. If you only measure one marker of inflammation, you really are not accomplishing much. And so it's important that I want you to understand a few things about C-reactive protein so that you can appreciate this particular marker, but it really is reflective of some of the other markers as well. And that, again, many of these inflammatory markers will be elevated in response to a whole variety of inducing factors. And so it's this non-specificity that may be useful not only in CRP but in other inflammatory markers. All of these inflammatory biomarkers have a function, okay? And CRP is derived from the liver. And so when you have an inflammatory response that the liver is responding by making this particular protein, this protein has a function. It's actually doing something. It promotes clearance of pathogens as well as damaged tissue and cells. And in the literature, you'll find that oftentimes you'll see this HS-CRP, and the HS simply is referring to the assay system that's used. It's a high sensitivity assay system where you can detect lower levels, and for healthy individuals, most publications will use the CRP high sensitivity assays. It is not a different CRP. HS-CRP is equal to CRP. It's the same protein. It's just that they're measuring it using a high sensitivity assay. So don't be confused by that. And so remember the level of CRP in the samples of the nurses with the trans fat intake, where it started at about 1 milligram per liter and went almost up to 2. And here you can see in a bunch of individuals who had surgery that in a matter of the first day or two after surgery, CRP levels went up to 13 milligrams per liter. And so eventually what happens is after surgery, people will recover and the CRP goes back down. And this you can see about day 13, the CRP is now back down to the normal basal level. It's this area of the homeostasis for CRP is what mostly this low-grade inflammation, this is where nutrition has an impact. It's not likely that nutritional interventions are going to dramatically change this diversion here, but it's how far inflammation goes down and how quickly it goes down. And again, you can expect in most studies that a significant portion of your healthy subjects will actually have undetectable levels of this biomarker as well as many other inflammation biomarkers. But CRP has been under considerable investigation by a variety of regulatory and other bodies to try to determine whether or not that it may be another risk factor or a biomarker of risk for cardiovascular disease. And again, the magnitude of the predictive value of CRP is nearly the same as changes in cholesterol content or blood pressure. And again, there's a variety of research bodies that are trying to validate CRP as a new risk marker Again, this has been going on for more than a decade, though, so not sure how soon or if ever it will actually be added to our armament. There's another wonderful report by the ILSI Europe group. This was a task force on nutrition and immunity, and this was published just 2 years ago, a year and a half ago actually, where they talk about various considerations for biomarkers for inflammation in nutrition studies. And again, I highly recommend this reading for anyone who's interested in this topic, because I can only superficially cover it. But the main points from this publication, or this report, is that there really is no consensus regarding which biomarkers of inflammation would be most appropriate to be measuring when it comes to trying to study the impact of foods or nutrients on health, but that all biomarkers should be scientifically validated and should be measured carefully, and that assessing the background or basal inflammatory biomarker levels may not be sufficient or are able to be predictive enough. And so there was a call by this task force that in fact what we may really need is to look for some way to look at stimulating inflammation and looking at how individuals responded differently, and whether or not people who were consuming different diets or following different dietary guidelines or patterns, how their immune response might be different or their inflammatory response might be different to a challenge. And again, this can be done in whole blood collected freshly from these subjects. And it is in this context that I was very excited to find a relatively recent paper that described really the first process by which you could comprehensively look at the inflammatory response from healthy individuals. And so this was out of the Milieu Interior Consortium at the Pasteur Institute. And they've done an excellent job of actually defining an assay system that is clinic available and that easy to use in the clinic. It's based on a syringe process. You don't have to actually do a lot of manipulation. You don't have to have a lot of training. And they use a whole host, actually 28 different inflammatory or immune stimuli, including bacteria, fungi, viruses, various specific agonists, as well as cytokines that are used in the clinic, as well as a couple of activators of T-cell immunity. And it simply involves collecting blood from the vein of a subject and aliquoting 1 ml aliquots into these syringes, which are essentially then capped and mixed with some cell culture media, and then you stick it in a 37-degree heating block on the lab bench. You don't have to have a CO2 incubator or anything, and you let it go for 22 hours, and then you collect the supernatant. You don't even have to have a centrifuge in the lab. All you have to do is put in a little filtration unit, separates out the cells, you get the supernatant, you stick it in your freezer, and then you ship it off to someone else, or you do it yourself if you have the capability of doing the multiplexing assay. And so again, it's another thing I wanted to very strongly suggest, is that the availability from a variety of commercial sources of multiplexing, of being able to measure anywhere from 10 to 25 different inflammatory biomarkers in the same samples across a wide range of concentrations, is a great technology which has really revolutionized our ability to do this kind of work easily. So I wanted to show you what the data look like. Okay, so this was 25 healthy individuals of European descent, an even split of, well, as even as 20, so 13 and 12 males and females. They were all about 30 years of age, 30 to 35 years of age. Again, these were healthy individuals, and what the data shown here is from just one of the 27 different inflammatory stimuli. This is endotoxin, also known as lipopolysaccharide. It is one of the most potent inflammatory stimuli that we know of. And here are some of, this is essentially all of the biomarkers that were used in this multiplexing, and I wanted to point out two of the more common inflammatory markers, interleukin-6 and tumor necrosis factor. The bars down here at the bottom are basically showing what the levels are in null, non-stimulated samples. And you can see that essentially there's almost no detectable IL-6 or tumor necrosis factor in the blood of these 25 healthy subjects. After the blood was stimulated with LPS for 22 hours, what you see here is the median and the 75%, essentially the quartiles, and then arranged to give you an idea of the kind of variance that exists. And so you can see these two inflammatory mediators, TNF and IL-6, they start out at virtually undetectable levels, and they increase by a factor of literally 100,000-fold. So that's pretty crazy when you think about that you essentially, again, in this situation. But I also wanted to point out, look how some of the inflammatory mediators, again, interferon gamma is a very common one. It starts out as undetectable, and yet you look at these 25 individuals and look at the variation there. So that would argue against using interferon gamma. That kind of variance in 25 healthy individuals would really create havoc with trying to demonstrate whether a particular intervention influenced interferon gamma responses. And so this kind of information is going to make it easier for us to identify maybe a few of the key biomarkers that we can use to target our future analysis for larger clinical trials, so we don't have to spend thousands of dollars doing all of these various biomarkers. And this also, I wanted to point out, is a different way of displaying the same data, and it indicates these are the particular inflammatory markers from these LPS-induced whole blood samples from these 25 healthy donors, and the largest induction occurred for IL-6 and IL-1 beta. And what I want you to see is this, each line and each color line represents each individual subject. And you can see here places where the lines sort of separate. So interleukin-1 beta, although it's one of the more important and it's more highly induced, there's a lot of noise here. And also interferon gamma, you can see how the lines are so separating like that. And then a really interesting finding is they found in these 25 individuals, 2 individuals did not make interleukin-1 alpha. Absolutely zero production. And this not only occurred for LPS, but every other stimulus that induced interferon alpha, not interferon alpha, interleukin-1 alpha. Interleukin-1 alpha is again an intercellular cytokine that plays an important role in the immune system. And they don't really know why these 2 individuals who are perfectly healthy but have some kind of defect in the ability to produce that. So that was an interesting finding. So I wanted to point out, so here's one of those individuals. So here's interleukin-1 alpha. You can see that individual, no matter, and down in this axis, this is all 27 of the stimuli, including the first one is the null, no stimuli. And heat-killed bacteria like E. coli give almost the same response as LPS. But what you can start to see is you can see that with different cytokines, inflammatory biomarkers, there would be patterns that would emerge across a variety of different immune stimuli. And again, the interesting question is, if you fed someone a healthy Mediterranean diet, would it change their pattern? Would it lower their pattern? Or would it increase their pattern? We don't know, but I think it raises an interesting question that I hope that future studies will actually help determine. So the advantages of this system is that this is a commercially developed and tested assay system that is designed for in-clinic use. So you don't have to have a lot of the tools and experience and expertise that my lab has, but in fact you could actually do this right where the blood is being collected in your clinical trials. So it's been designed for that in mind. And that they use a diverse immune stimuli that have been optimized and quality controlled. And again, one of the key points about this particular system that I value very much is that they identified and did dose-dependent or concentration-dependent studies with each of these immune stimuli, and they've chosen concentrations that are submaximal. And this is very important because you can at times overwhelm nutritional or other interventions by throwing so much of a stimulus at immune cells, or at, in this case, at blood cells. We've seen this time and time again in our laboratory. And so using stimuli at concentrations that are submaximal actually enhances the sensitivity of the assays. The biggest disadvantage at this point is this is a brand new commercially developed operation. They've got a lot of costs that they have to recoup, and so the out-of-pocket costs for the first few years are probably going to be pretty expensive. And actually, I don't even have a cost that I can quote you, because the company said they wouldn't tell me what the cost was unless I was actually going to be wanting a quote. So I can't tell you, but it's going to be thousands of dollars per subject right now, and that's that's prohibitive. So there's a lot of information about and talk in this field about inflammation, that dietary endotoxemia or post-prandial endotoxemia. Endotoxin, again, is the word that we also identify as lipopolysaccharide. It's on gram-negative bacteria. It's a component of the cell wall, and it is one of nature's most potent immune stimulators and inflammatory stimulators. And again, we know, or at least we think we know, that endotoxin from our gut microflora are absorbed in response to a high-fat meal. And yet, the rodent studies tell us this quite clearly. The human studies are more uncertain, unclear regarding whether or not endotoxin is actually being absorbed, and if it is actually inducing the inflammation that is associated with these kinds of excursions. There's a lot to be learned here, and I would recommend that if you're interested in this topic, that this review article on this topic in the Nutrition in Clinical Practice in 2012 by Kelly and co-workers. It was really an excellent paper that described some of the limitations of what is known and not known. And so the real problem is that the way in which any laboratory can assess whether endotoxin is present in plasma or serum from human subjects, it's really problematic. We do not have a good, reliable way of doing this, and yet there are dozens of publications claiming to be reporting endotoxin levels, and yet we're not really sure we should trust those values. And again, it's the most common assay that's done. The limulus amoebocyte lysate assay, the LAL assay, is notoriously unreliable. And again, the biggest problem is that it doesn't distinguish between non-toxic or non-inflammatory forms of LPS and the inflammatory forms of LPS. And that's been well documented more than a decade ago, and yet it's routinely used to measure the presence of LPS. And what's interesting is we're just discovering that an enzyme that we weren't quite sure what its role was in terms of our digestion, this intestinal alkaline phosphatase, in fact, is a critical enzyme that's expressed heavily on the endothelial cells lining the mucosa of the GI tract. Its primary job is to dephosphorylate LPS, which essentially makes it non-toxic, non-inflammatory. And then in addition to that is that there's all kinds of proteins as well as free fatty acids that can interfere or cause false readings in a lot of these endotoxin assay systems, and so that's very problematic. So this is a depiction of what lipopolysaccharide looks like. It's a polysaccharide. It's got carbohydrate on it, and then it's got this lipid component, these 6 saturated fatty acyl groups, and then these 2 phosphates on it. And this is part of the structure of the membrane of gram-negative bacteria. And again, if you substitute these saturated fatty acids with monounsaturates or polyunsaturates, this is no longer pro-inflammatory. And again, this is not dietary. This is not a dietary thing. This is the genes of certain bacteria will put monounsaturated fatty acids in it, and those bacteria will not elicit an inflammatory response. And I already mentioned that, again, in the lipid A portion, which is the inflammatory portion that triggers inflammation, removal of one of those phosphates by intestinal alkaline phosphatase is all that's needed to essentially completely obliterate the inflammatory. And then saturated fatty acids, particularly those that have a similar carbon number, peaking right around 12 carbons long. So saturated fatty acids of those length, or 14O, are very good at stimulating inflammatory responses in cell culture. And we just don't know whether or not that really translates into in vivo when you consume fats that are rich in 12O or 14O. Are those inducing the same response? But it really brings me to the last new topic that I want to talk about, and that has to do with, again, certain fatty acids do seem to have an effect. We've already seen data that trans fatty acids promote low-grade inflammation, and certain similar data exists for saturated fatty acids. Most of the data for monounsaturated fatty acids suggests they're neutral. Sort of sounds familiar to the cholesterol story. But what about polyunsaturated fatty acids? So linoleic acid is the most common PUFA in our diet, making up anywhere from 5 to 10 energy %. And there's been a message that's been going out for the last 10 years or so, suggesting that the use of vegetable oils, most of which have 50% or more of their fatty acids are linoleic acid, is promoting inflammation through a process by which tissue linoleic acid is increasing tissue arachidonic acid. Arachidonic acid can be metabolized to pro-inflammatory eicosanoids like prostaglandin E2 and LTB4. This elevates inflammation, and this adds to these chronic diseases where inflammation plays a role. So that's been the story, and again, seems perfectly logical. In fact, I believed it for a number of years. And then I got asked by an excellent food scientist, Guy Johnson. He said, Fritsche, I need your help. I need to do a systematic review of the literature on linoleic acid and inflammation. And I said, well, okay, fine. I expected that we'd find all kinds of evidence in the human literature that would show that consuming a diet high in linoleic acid would promote inflammation. Of course, it makes sense biochemically speaking. And in fact, so we spent a year and a half doing the systematic review. I don't know how many of you have done one. I would recommend that you avoid it at all costs. But if you are stuck and you do it, okay, so one of the things that we had to search the literature and find opportunities for subject groups in which dietary linoleic acid concentration only varied relative to oleic acid, the monounsaturated fatty acids, with no significant differences in saturated, trans, unsaturated fatty acids or omega-3s. Most of these human studies were actually omega-3 studies, where we used one or more of the different control groups in the fish oil, not the fish oil fed groups, but the control groups, and we were able to do the comparisons among different control groups within the study. So these studies were not designed really to address this question. Of the 15 clinical studies that we identified, 5 of them looked at CRP, and this is just representative data. And again, so here's the 5 studies, and this is the initial linoleic acid intake, grams per day. You can see that the dietary recommended intake for linoleic acid is somewhere between 11 to 17 grams per day. And so this is sort of in the low end of normal. And you can see the huge increase of linoleic acid that was induced in these clinical trials. They really were pushing the limit. And the change in CRP in milligrams per liter, this is the change, not the actual concentration. In one study, 2 different groups, there was a small increase, non-significant. But in the other studies, it all went down instead of up. It went down. None of this was statistically significant, though. This data, along with some of the other data for other biomarkers of inflammation, all led to the same conclusion, and that is that there was no scientific evidence that in humans that consuming higher levels of linoleic acid in a diet was associated with an elevation in circulating concentrations of a variety of inflammatory biomarkers. So why? Why are we misled? So unfortunately, my slide got messed up, but so linoleic acid and arachidonic acid metabolites play a role not only in inflammation, but the missing magical word here is that they play a role in resolution. So this shows all of the pro-inflammatory roles for linoleic acid and arachidonic acid as we understand them. But in yellow, I've indicated that these same fatty acids also they are a source of anti-inflammatory or pro-resolving lipid mediators. And it's the relatively new discovery of these lipid mediators. And I'm sorry that the slide is so confounded. It's a mess. So, but in any case, this is in a publication that is also going to be cited in this report. So I'd be happy to share this with you. So the main point is that it may be the reason why we're not seeing increased inflammation is because of the fact that these things they don't just make pro-inflammatory mediators. They also are sources of potent pro-resolving mediators as well. They just simply may be being missed or not measured. So fatty acids and low-grade inflammation, when substituted for sugars, saturated fat, or trans fats, N6 polyunsaturates tend to actually reduce low-grade inflammation. Omega-3 fatty acids inconsistently, or if it does, it only modestly lowers inflammation in humans. And that the response in mice is very impressive. You can almost treat or cure or prevent any inflammatory disease in a mouse with omega-3 fatty acids. And the reason for this is because mice are hyper-responsive to the anti-inflammatory actions of omega-3s. Their immune cells concentrate much more omega-3s than immune cells of humans or other animals. And so it makes the mouse a very powerful model to get publications, but it doesn't necessarily help us translate what omega-3s do in humans. So a word of caution. The real story about omega-3s is just emerging now, I would say, and that is that omega-3s, as well as some other nutrients, may actually be playing an important role in resolving inflammation. And advances in lipidomics are helping us understand this particular role. And I would encourage any of you nutrition researchers who are interested in asking questions about inflammation is design your experiments to measure the resolution phase of inflammation, not just the peak inflammation. Measure how quickly inflammation is being resolved, because that is, I think, where nutrition has a potential role to play, more so than affecting the actual onset of inflammation. In lipidomics, the Lipid Maps is a consortium of a variety of labs across the country that are doing marvelous things and that are sharing open source information about methods as well as structures for a whole host of bioactive lipids that occur in tissues and in the blood. And again, who knew that there were over 5,000 fatty acyl metabolites that we needed to pay attention to? I certainly didn't. I probably could have come up with about 100 in my head myself. So again, it's absolutely amazing the diversity of what lipids exist in nature. And again, it's being answered in a systematic way. And I would encourage you to collaborate with people who have the expertise to do this and add it to your clinical trials. So for me, this is my bottom line message. We need better methods for quantifying endotoxin in human plasma and serums. And until we get that, we're really going to continue to flounder in this part of the inflammation story. CRP and other inflammatory biomarkers need to be validated and for their ability to predict health outcomes in disease. We continue to need people to measure as many inflammation biomarkers as possible, both before and after interventions, and possibly even intermediate stages of your studies. Suitability of ex vivo immune-stimulated inflammatory profiles should be investigated, again, but the cost needs to come down in that for that really to occur. Again, I strongly suggest you use multiplexing. There's a variety of commercial sources available for those kinds of kits, and they're fairly easy to use, and they're fairly robust. Lipidomics, not everyone can afford to do this. Not everyone has the expertise, but there are centers across the country for doing that, so I would suggest that you incorporate some analysis some of your samples to be sent off for that analysis if you're working in this field of fats and fatty acids. And then finally, again, the inflammation story is the beginning. Think about the end. The end is resolution, and that is where nutrition is likely to have an important impact. And so we should not lose sight of that, and again, design your experiments and your sample collections to take that into account, because I think that's where the undiscovered territory and greatest benefit of nutrients and nutrition is going to occur down the road. So thank you very much.